cell and make it into a heart cell, it goes through a whole series of different processes. The cells have to move around, they proliferate, they differentiate, and what I'm really going to talk to you about is this really amazing process of rhythmic contraction. And the process by which this occurs is relatively simple in the sense that one of the ways that we do this, and these are mouse cells, we also do this with human cells, but here you can actually, what we do is we actually put them in a controlled environment. And in this case, it's a hanging drop method where we take the cells and we put them into a hanging drop, just like the drop of water that would be, say, on the dew of a tree or something like that. And then the cells actually come down to the bottom of this area, and in a relatively controlled setting, they start talking to each other, and then they start saying, okay, well, let's make a ball, which is sort of like an embryo, and then they actually start beating here, say, on one side, and then there's other cells that are here that we don't have markers for because they're not moving, but these cells are actually indeed neurons and blood vessels and all sorts of other cells that if you watch them grow, that you could do that. The interesting thing about this is that although this is not a mouse and then the other ones are for humans are not humans, that these will respond though to the same hormones and drugs that you do give to people. So for instance, you know adrenaline. Adrenaline is what you get an increase of when you almost get in a car accident, or when the Neanderthal saw the lion. It would be is you increase adrenaline, that increases your heart rate, and that's exactly what happens here. We measure the heart rate of these little balls of cells, and they increase, and then the exact same hormones that slow your heart rate down do the same. What's nice about this compared to the mouse or to a human is in fact that you can also do quite a lot of other things to them. So you can, for instance, you can cause beating. You can actually mark the cells in green fluorescent protein. You can see that the beating cells are green and vice versa. You can make lots of other cells. These are just some neurons. I just wanted to show you some of the other cell types that show up in here. We can, in fact, the cells, I saw some nicely beating cells there, but then you also have these cells which are beating in an irregular fashion, and you have two different pacemakers, as it were, in this particular culture. So there are different types of muscle. One of them is there's cardiac muscle, which beats rhythmically and it beats throughout your entire life, and then you also have smooth muscle, which actually lines your lungs and your intestines and your blood vessels, and that has a contractile process, which is very different. You can see, you don't have to be a cardiologist or physiologist to see how different this is than the beating of the cardiac cell. And actually, there's a cardiac cell right over here that's blinking like this, but you have this new smooth motion. We also can make skeletal muscle, and you can see this here is a plate of cells which actually became all skeletal muscle. So you can, and there are obviously diseases of skeletal muscle like muscular dystrophy and so on that would be really very useful for therapy. The simple differentiation is not something that's limited to basic scientists. We actually taught all of our techniques for differentiating cells to the people at the Exploratorium. They actually grow the mouse embryonic stem cells, and they'll be growing human iPS cells in this imaging station here in San Francisco. And you can go and interview kids who are looking at microscopes, looking at stem cells here at the Exploratorium. And like I say, we'll be actually deriving iPS cells there, so that'll be an interesting process. So now, how do we know that this actually is relevant? You got a little beating cell at the bottom of a dish. What does that mean? I showed you they speed up and slow down. One of the things we can do with mouse stem cells that we can't do with humans, human stem cells, is we can take the stem cells, we can see a phenotype. For instance, here we had a mutant, and the name of the gene is unimportant, but we have a mutation that causes an irregular beat rate and an irregular response to a drug. And we can take that mouse stem cell and make a mouse out of it. and we can make a mouse that now has the exact same mutation as what was in there, and that actually, that phenotype that was in the stem cell reproduces in the whole animal. And we were able to do that not only for beat rate and how it responds to certain hormones, but also this beat rate variability, which is something we didn't even believe would be something that we would see in a plate. But heart rate variability is an important measure, actually, of heart health in general. And we were able to reproduce that in the stem cell, and the stem cell-derived tissues, as well as the whole animal. Using that data, we were actually, from about this particular gene, we were actually able to then identify a human phenotype with there are human variants of that gene, and we were able to make this leap. Now this leap is actually a huge leap, because a mouse heart rate beats at 600 beats per minute, and a human heart's, generally speaking, is much, much slower, more like 80 or 90. And so we were able to make that leap, but it's really, as was just Nisa said before, we're not mice, and so this is a big, big leap. What we would like to do now, and what we're able to do now, really, is now take, since we can't make stem cells into people, we can now take people with a phenotype, and just by reversing the arrow, essentially, we're able now to make stem cells, and with the induced pluripotent cells here. And I should mention that Shinya Yamanaka, who really identified this technique and pioneered it, is essentially is a California story onto his own. He did his postdoc here, just a few miles from here at Gladstone and UCSF. He learned about how to culture mouse embryonic stem cells here, and he really brought that technology back to Japan, and then actually took it a step further by learning how to do the reprogramming, and that is really a terrific, really local story, and he does have a lab here about 25% of the time. What we're doing in terms of how we're actually thinking about this process, so how, what's the phenotype, and certainly I think the neural phenotypes are very interesting, but given the fact that these stem cells actually beat, and we knew that the phenotype actually relates from the stem cell phenotype to the mouse phenotype, we've been focusing on cardiac problems and beat rate changes. And what the initial thing that we're focusing on is called long QT syndrome. And all of you know what an EKG looks like in general, and all of these little waveforms are named. This is the Q wave here, QRS, and then here's the T wave here. And if the QT interval, as shown here, is longer than normal, you have a funny looking EKG. And that is actually, doesn't seem like much of a problem, except for the fact that people who have the long QT, the mutations that cause long QT, drop dead. And they drop dead suddenly, and they drop dead when they take certain drugs, many drugs, which you can buy at the regular drugstore. And so this is an EKG of somebody who actually did die for a little bit, fell on the ground, and then they came back to life, fortunately for that person. For every person who comes in the emergency room who has sudden cardiac death, there are actually many relatives who don't even know that they have the disease. So in other words, the clinical prevalence is 1 in 5,000, but the genetic prevalence is much more. So there are families such as this, and we collaborate with a family in Finland for this particular family, but there's plenty of families around here like this, where this person died of sudden cardiac death and was brought into the hospital, but when they have very good records, and they do something really incredible there. They actually have a healthcare system where they take care of their citizens. I don't know, it's weird. So they could bring all their people in. They didn't actually worry about insurance. They could actually put a test out and say, sequence the known genes for long QT, and they sequenced the entire family. The entire family came in, and you have here, now these are affected individuals, and they have a funny EKG, right? You can actually see them on the EKG. But interestingly, these people over here, they have the gene. If they took, for instance, erythromycin, they could drop dead. And a long series of genes, drugs, they could drop dead. but they had no idea, and even if they'd done an EKG, they would never know that they had this problem. And so this is really, this is very valuable to them, in particular for these people, because they can actually take a drug, just a beta blocker, and for these people, they can actually be protected. Their rate of dropping dead goes actually down, but there are actually 10 different types of long QT syndrome for which there are drugs only for, good drugs for only one of those 10 types. So what would be really interesting now is to make iPS cells from this person and this person, because these EKG is abnormal, but this one's normal, and now to see whether or not we can take iPS cells and from these individuals and see if they are different indeed. Now, this raises a problem, because how do you do an EKG on an embryoid body? People, we understand we can put the electrodes on, but actually, how do we actually get that? And the technical hurdle here is we know that we're able we know that we'll be able to record this, but how to do this in a robust, high throughput way so that we can use it, for instance, to identify drugs that correct the long QT syndrome, that we can use it to screen for drugs that would cause long QT syndrome is particularly important. I should add that long QT syndrome, although it's really bad for the people who have it and drop dead, it's also bad for the drug companies, because the drug companies, if you market a drug and you put it out into the to the public, and it's giving to millions of people, and then it actually has this very bad side effect, that ruins essentially the market for the drug. And so they're very focused on identifying drugs which would making sure that their drugs do not cause long QT syndrome, especially in these patients who have the defined disease, but also in patients who have known side effects in the past and actually no known genetic risk factors. And we can potentially make iPS cells from those people as well. So the technical hurdle for us, the making the iPS cells, actually has been relatively straightforward. It's been straightforward in multiple labs here in San Francisco, and I think that probably in San Diego as well. That we've found it to be remarkably robust, in the sense that rarely, for us at least, have we ever gotten a protocol about something complicated and then found that it was easier or more robust than was reported. And that's, for us, in our experience, that's been the case, at least for the initial IPS technique. So doing this experiment is, in terms of making the iPS cells, is relatively straightforward. It's the analyzing. How do we do an EKG in these cells? That's going to be the problem. How to do it in such a way that we can actually tell the difference. Now, I remind you that we can't do the experiment of taking this long QT carrier and giving them a whole series of drugs until this happens to them, right? This is an experiment of nature. But hopefully what we can do is take their cells and put them in a dish and give them a series of drugs and essentially induce this effect. And so this is actually a model of a recording on an individual cell that's going on during this EKG. And this is how we actually, when we measure the channel activity in a cell, we imagine that the carrier, as well as the person, in other words, the person who has the bad gene, the long QT gene, and their normal relative, will probably have a relatively similar recording when we first get the iPS cells. And the trick now is going to be to optimize the conditions, to identify the conditions where we can tell the difference between the individual that has long QT syndrome and their normal counterpart. We can do lots of different tests to bring these conditions out, and these two. The only reason that we would know that this is really important or valid is that we'd have the patients. And the patients are different, and we know that the patients are different because of their various response to drugs. And so we can match the patients up to each other, and we can say it's clinically relevant that this particular drug, for instance, causes this change in this person but not that person, because we know in people, when we give a drug, this same sort of change happens. And so this would be as taking we basically have a cellular-based assay, and we're using the the people essentially as a yardstick to say this is a relevant change versus this cellular change may be totally irrelevant because actually it's different, but it's different in a way that doesn't really match up to the genetic phenotype. So in closing, sort of the future applications, from the basic science point of view, stem cell-derived myocytes will provide a powerful new platform just to study how heart rhythm happens. This is a process that it happens every day to you thousands and thousands of times, but we don't understand the basic mechanism of how it happens. And this is we can get sort of basic scientists, computational scientists, people like that involved in this exciting process. But also it allows us to make iPS-derived myocytes can be used to better understand the cardiac genetics. And so there are many different types of mutation, some of which are known, but maybe this will be a platform to understand some of those that are unknown. And then a robust long QT assay could be used to develop new drugs. So there's, like I say, there's types of long QT for which there really are no good drugs for, and rather than testing drugs out in these people, we can test them at least in a plate first and try to identify those. And then finally, the iPS cell-based long QT assays could be used for safety screening of new drugs. So before a drug is released out into the public, it could be tested on this. And I should say that the current state-of-the-art for this is actually to take a hamster cell and transfect it and force it to be expressing a particular type of channel, it's called the hERG potassium channel, and then testing drugs on this. This essentially hamster fibroblast is a good test, but it's not a human heart cell. And so having human heart cells, which we know that we have an assay that we've compared to essentially, which we've used human genetics as a yardstick to say it's clinically relevant, hopefully will make the drug supply safer for a future generation. So that's all I wanted to talk about. Questions?
Yeah, excuse me. So we were talking about doing, you said on one side, it's, one thing that's unknown is how you actually do an EKG on it. I called it embryoid body.
Embryoid body.
So is the embryoid body the iPS-induced, it's after you've gone back, you've taken the cell and through iPS, you've brought it back up to a certain undifferentiated level, but it's still a heart cell? I was a little confused by that.
Okay, so what is an embryoid body, I think is the question, which is a terrific question. And I think this is probably the best example, at least a picture of it. The embryoid body is the entire group of cells. These were essentially a single type of cell. It could have been one cell initially, but usually we start with about 500 cells that were all essentially identical or indistinguishable from each other. We allow them to congregate together, and then they begin to differentiate. And that differentiation is the embryoid body. So it's embryo-like, embryoid body. And this embryoid body is this entire mass of cells, which started off as 500. It's probably more like about 5,000 cells here. So it's probably gone through a few doublings. And you have here fibroblasts growing out. You probably have neurons, and you have a tiny area of beating cells. This, in fact, if we just isolated the cardiac cells, it's probably just a little group over here. But because they're actually contracting, they're actually pulling the tissue around them. So they're kind of like, waving themselves, basically, that they're actually doing the beating. But the entire thing is called an embryoid body, if that answers the question. And then I think you had a question about recording, or is that?
So, sorry, say again.
So it can come from both iPS or ES cells.
Right. Any kind of, and going back to Dr. Goldstein's talk, this is a pluripotent stem cell, so it can make any tissue in the body. So if this is a human cell, it can make any human cell in the body. The only reason it's not totipotent is because it can't make placental tissue. And so we can't find placental tissue, which is on the outside.
So theoretically, you could get there through iPS, but did you say you have, or you said there's labs that have gotten to this point
We've
already?
got this, we have, you know, I do have movies elsewhere on this computer of human iPS cells that are beating just like this. It's the same thing, essentially.
But
It's not any...
Those 500 cells that started down this road, those 500 cells, are those from embryonic stem cells, or those iPS? Where are those 500 cells?
So when the 500 cells that are start out, it's really, as far as we can tell, the iPS cells and the embryonic stem cells that are pluripotent are essentially are equivalent with respect to their ability to do the differentiation. Each embryonic stem cell is different than other embryonic stem cells, and so there's a diversity within embryonic stem cells, and there's a diversity within iPS cells. And as far as we can see, that they are overlapping clouds. Now, one iPS cell could be quite different than one embryonic stem cell, but that doesn't mean that the clouds are not overlapping. And as far as we only have a few data points in the clouds of both of them. We have very few embryonic stem cells, we have very few iPS cells, but that's where the bet is right now. And as Dr. Goldstein mentioned, we don't really, we're somewhat interested in these iPS cells because they have, you know, because they have this potential, but the ones that we're really going to be most interested in is the ones that are made without gene integration. And those are, you know, those will be coming, we don't know exactly when, but when they do come, those will be really identical because there'll be no gene integration and so on.
So just for clarification, can you get to an embryoid body from an iPS?
Yes,
Okay.
absolutely.
Yeah.
You can, everything that you can do with an embryonic stem cell, you can do with an iPS cell with respect to sort of the basic biology experiments, as far as we can tell. There's no, and the gene expression profile and so on.
It seems to be sort of an article of faith that what you just said, that we will get there, that you will be able to substitute, you know, a drug or something for the genes that are used to generate iPS cells. And I wonder if you could explain why that's such an article of faith among scientists.
Well, I think that
among some scientists.
there are just so many ways to this answer. Some of them are hard, some of them are easy, but there are so many ways that you can to get this. So the bet actually is that there'll be 10 or 15 ways to do it, and you know, you can put in genes so that they're. I mean, all we're talking about is putting in 4 genes. So you can put in genes so that they're not integrated, and so that they can be removed. And that's sort of the very most straightforward. You can put proteins. You can take the genes and code proteins. You can take proteins and just stick the proteins in directly. Theoretically, I mean, none of these experiments. I'm not talking about things that have been done, but I'm just saying that there are artificial DNAs, and for every protein that exists out there, every kind of network that we talk about, there's both an activator and an inhibitor of that. There's lots of ways to inhibit proteins at the protein level, or knock down genes by killing genes by or knocking down the RNA level with artificial RNAs, that inhibitory RNAs. And so if you can kill the inhibitor of something, then that's essentially like activating. And so within any of these pathways, you should be able to do that. So there's right now there's an embarrassment of riches within for which a cell biologist has tools to attack this particular problem, and mimicking 4 genes is not, it's difficult, but it's a very, very solvable problem. At least that's my own. And you know, if anyone wants to bet against me, then I've got, you know, I can do it right now.
I think we should move on, and we should also say that CIRM is funding someone who's using small molecules as a way to mimic the genes.